And we're live.
Welcome, everyone. We're going to get started in just a few minutes. Thanks so much for attending the FASCIB webinar on COVID-19, the critical role of non-human primate research. Just sit tight and we will get started at the top of the hour. All right, it's the top of the hour. Let's get started. So welcome everyone and good afternoon, good morning for those of you who are joining us on the West Coast for the FASCIB webinar, Combating COVID-19, the Critical Role of Non-Human Primate Research. So I'm very excited to be with you here today to dive into this topic. My name is Naomi Harlem-Bacchus. I am a Senior Science Policy Analyst at the Federation of American Societies for Experimental Biology, or FASCIB. And I am very excited to dive into this topic today because not only is it important for us as we try to continue to navigate this ongoing pandemic that is COVID-19, but it's also really underscores just how instrumental animal research is in helping researchers understand this disease and help advance scientific advancements and treatments and vaccines. So as many of you know, companies like Pfizer and Moderna have recently released early vaccine data showing the efficacy of COVID-19 vaccine. And those results were possible because of animal research. And we are grateful to that. And today we're going to learn a little bit more about that process and what scientists are working on. And as we speak, the FDA advisory board is actually looking at the Pfizer vaccine to determine whether or not it will receive emergency use authorization. So today we'll explore a little bit more about non-human primates and specifically look at how COVID-19 is conducted at the National Primate Research Centers across the US. And I know we're all experiencing some Zoom fatigue, but I hope this webinar will be informative and allow you to gain a better appreciation for the critical role of non-human primates. So just a few housekeeping things. The webinar is being recorded and it will be posted on the FASCIB website within the next few business days. So the PowerPoint slides will also be available. With that being said, at the end of the webinar, let's make sure that you guys are connected to audio. I forgot about this slide. Please make sure that if you're having trouble with audio to select the correct speaker system and make sure it's coming through where you would like it to. And at the end, there will be time for questions and answers. Just if you see the box at the bottom, the Q&A, type your question in the box, hit send, and we will receive it on our end and go through them as we receive them. If we don't have enough time to go through all of them, I will collect all of the questions and answers and distribute it to the speakers, and they can answer in their own time so we can have it as a resource on the FASCIB website. So I am grateful also today, before we get started, I want to thank my colleague, FASEB Science Policy Fellow, Dr. Tianlu Ma. She is the FASEB webinar that you see on your screen, and she's going to be playing tech backup and watching the questions and answers come through. So because this webinar is running smoothly, I have to thank Dr. Tianlu Ma, and I have to thank our two speakers. I am delighted to introduce to you today Dr. Jay Rappaport. He's a professor of microbiology and immunology at the Tulane School of Medicine and is the current director and chief academic officer of the Tulane National Primate Research Center. And Dr. Nancy Haigwood is a professor of pathobiology and immunology and the current director of Oregon National Primate Research Centers. Dr. Haigwood also has 17 years experience in the biotechnology and pharmaceutical sector, so her unique perspective will be crucial for today as well. So without further ado, I'm going to turn it over to our first speaker, Dr. Jay Rappaport.
Thank you very much, Naomi, and also Tianlu. It's very exciting to be here, and this is an exciting and important topic about how non-human primate research is contributing to our efforts against this COVID pandemic. The non-human primate centers, the NPRCs, the National Primate Centers, are working very closely together in developing protocols and harmonizing studies. Nancy Haigwood will talk a little bit about that, or talk about that in her talk. What I'm going to be talking about is some of the studies that we're doing here at Tulane and present some of the data. We're doing some therapeutic studies, some vaccine studies, animal model development, and some passive antibody studies. Some of the studies are under confidentiality agreements, but I'm going to talk about some of the studies that I can talk about. One is a pilot study we did, and basically I'll present results from 3 papers. One from Robert Blair on the pathology, one from Monica Vicari on the role of monocyte macrophages and cytokines in the disease, and one from Tracy Fisher showing CNS pathology. And additionally, we're doing some studies with Operation Warp Speed using monoclonal antibodies, and I'll present a little bit of that work as well. Can I have the next slide, please? So this study was our initial study that we did. It was a pilot, and we did a study on 4 Indian Rhesus macaques and 4 African green monkeys. So we had 8 animals total. These animals were a little bit older. The African green monkeys were approximately 16 years old. They were wild caught. The Rhesus macaques were 13 to 15 years old, and they were bred on site here at Tulane in our breeding colony. These studies were done to look at the effect of COVID-19 in this model. In each species, we infected 2 animals by different routes, either the aerosol route or intratracheal, oral, intranasal, and intraocular. We collected samples pre-exposure and also post-infection at various intervals that are shown here at this slide. We collected blood, urine, stool. We did physical exam, bronchial lavage, and we detected virus by bronchial brushes, mucosal, buccal swabs. We collected CSF, and we also did PET CTs, chest X-rays, and plethysmography. Can I have the next slide, please? The first paper that I'm going to talk about is now in press in the American Journal of Pathology. Should be coming out very soon. And there's many authors here, and this is really the work. Takes a village to do these studies that are done, in containment, in biosafety level 3 facility. We have a core team that does work in BSL3, and also we have a core animal care team. We have our veterinarians. We have people doing these various assays. So next slide, please. So this slide shows some of our results, looking at how much virus is coming out, in these animals, looking at swabs. The buccal swabs are like in the cheek pouch area, nasal swabs, pharyngeal swabs, bronchial brushes. Even some of the females, we showed virus in the vaginal swabs. And also there's a good amount of virus in the rectal swabs, although with the kinetics appears to be a little bit different. One of the things that we really noticed from this initial study looking at these viral loads is that there wasn't much difference between the kinetics of virus appearance in the African green versus the Rhesus macaques, although the Rhesus macaque, you know, if you can look in the bronchial brushes, the Rhesus tend to resolve toward the end of the study, where some of the African greens seem to have you know, persistently elevated viral loads. But there's not much difference in the level of virus produced, and overall there's really you know, there's not that much difference in the species. Next slide, please. This slide shows some of the pathologic changes in COVID-2 exposed African green monkeys. And what you can see here is an animal where you can see 22 hours before the appearance of disease. What we call, this was actually acute respiratory disease syndrome. Here you can see the lungs are clear, and on the right side, 22 hours later, you can see this ground glass opacity, which is really distinct from, you know, from pulmonary pathology characteristic of ARDS. And when you look at the gross pathology on the lungs and necropsy, you can see there's this area of what's called consolidation. And this is representative of considerable edema and fluid accumulation. These lungs actually in necropsy are really not collapsible. So considerable amount of fluid and disease in the animals that had ARDS. Next slide. So this slide shows what's happening by histopathology and a little bit of immunofluorescence data. At the top two panels, you can see at low power and higher power what normal lung looks like. And these are H&E stains. But you can see that this animal at time of necropsy, the pink is really fibrant staining that stains dark pink with the H&E stain. At higher power, there is considerable invasion of inflammatory cells. These are monocytes, macrophages, neutrophils, T cells. You can see here, high power, there's a multinucleated giant cells, and these are really extensive inflammation is occurring here. In the green, you can see that this stains positive for the CoV-2, the SARS-CoV-2 nucleoprotein, as evidence that there is infected cells here. There is some red staining that you may be able to pick up and see, and this is picking up the ACE2, the receptor for SARS-CoV-2. Next slide, please. So the lung injury that we see in the rhesus, I showed you before, the pathology was more significant in the African greens that had ARDS. But if you look in the Rhesus macaques, at time of necropsy, there was considerable resolution, but there is some remaining pathology that I'll be able to show you. But this is just going to show you what you see at 7 days post-infection compared to normal lung. And you can see again, there's quite a bit of this pink staining, fibrin deposition, and at higher power, you can see the extensive inflammatory cell infiltrate. Next slide, please. So even at 30 days post-infection, and by this time, the infection is really nearly completely resolved, but nevertheless, histologically, you can see that there remains some damage that is apparent when you look at these lungs. On the top right, you can see this pleuritis with these inflammatory cells. These are inflammatory cell infiltrate. There's fibrosis, this pink area. There's fibrosis here as well. There's scattered foci of this pulmonary infiltration of inflammatory cells and fibrosis. So despite the fact that the infection is resolved, there is damage that seems to persist. And this is I think, very concerning when we think about people who have, let's say, convalesced from COVID infection, and there may be, considerable remaining damage that persons who are infected have to contend with. Next slide, please. This is a study that was done on the same group of animals and the same pilot by Monica Macari's group here, the first author, Marissa Fahlberg. And this is looking at the cellular events of acute and resolving progressive COVID-19 infection in the non-human primates. Next slide. So here what you can see is on the left, we see what was going on with cytokines in the African green monkeys, and on the right, you see what's going on with cytokines in the Rhesus macaques. The pattern is very different here between the two species. At early times, at the 7 days post-infection, you can see the African greens have fairly large increase in interferon gamma. And then when we look at at later times of infection, at time of necropsy, there are many cytokines that are activated here. And this is what we call cytokine storm. There's actually 3 of these animals, 3 out of the 4 African greens, I would say, had cytokine storm. And 2 out of 4, the ones that are in the box, had exhibited acute respiratory disease syndrome. This is very different than what we see in the rhesus, where early on there's really not significant, in cytokine activation. Particularly, you don't see this interferon gamma activation. Although at latter times, you do see some of these other cytokines being activated. For example, in a couple animals here, or a few, you have IL-1 beta activated, and then you see IL-10. Next slide, please. This shows chemokines and plasma levels in CoV-2 Rhesus macaques and AGMs. And here they're basically arranged from most severe disease on the left to less severe disease on the right. There's something wrong with the fonts here, but one of the cytokines that's very important here is TARC, or CCL17, which appears to be very largely increased in the animals with severe disease. There are other cytokines that are shown here. Many of these, or the majority of these, are cytokines that are associated with myeloid cells, MIP-1 alpha, MIP-1 beta, MCP1, etc. Can I have the next slide? This shows what's going on with monocyte dynamics and trafficking. If you look at viral load here, and these are with bronchial brushes, there's a very nice correlation between the absolute number of monocytes that are in circulation and at week 1. And it looks like with increasing amount of virus being produced, there's increased numbers of monocytes in the circulation. I mentioned how TARC, or CCL17, was important. The absolute number of monocytes correlates very closely with the level of TARC in plasma. I mean, this correlation is extremely significant. Now, looking at subsets of monocytes, one of the subsets that Monica's group was interested in was the non-classical monocytes, or CD16 positive monocytes. And these are thought to be involved in this like an M2 phenotype or immunosuppressive, and have maybe suppressive effects in promoting Th2 responses. And you can see what happens, and these are normally about 5–6% of total cells in PBMC. Here you can see that this population, the non-classical monocytes, CD16 positive monocytes, decreases at week 1. They seem to leave circulation, and then at week 2 and 3, as the infection resolves, they come back. And this shows what's going on in both the African greens and the Rhesus macaques. Now, when we look at what's going on in the bronchial lavage, something very differently is happening. Initially, at baseline in bronchial lavage, there is no cells. These cells are not detected. But at week 1, when they leave the circulation, when they're no longer present in circulation, they appear in the lungs, in the bronchial lavage. So they're trafficking out of the circulation and into the lungs at week 1. So this is really what's going on here. And then these cells sort of disappear at week 2 and 3 as the infection resolves. I have the next slide. So this last of the 3 papers that I was telling you about, this is by Tracy Fisher's team here. And here she's looking at the neuropathology and the virus in the brain in SARS-CoV-2 infected primates. And this is in revision for Nature Communication. So here you can see in this top left panel, this is a section in the cerebellum, and the arrows show areas of microhemorrhage. And here you can see basal ganglia. You can see these also. And what's evident here is when you see the lumen of the blood vessels, that the red blood cells are really on the wrong side of the lumen. This is because the vessels are hemorrhaging. So this is something that's happening in the brains of animals that have SARS-CoV-19. And there's really significant pathology. This is also basal ganglia. Here's some section in the brainstem. So this is something, it's becoming evident that persons with SARS-CoV-2 infection are getting neurocognitive impairment, and some patients are called long haulers. They're taking a long time to recover. There was a study by Igor Koralnik's group that showed probably about a third of patients with SARS-CoV-2 had some evidence of CNS disorder. And I think this could be a mechanism responsible, for what is happening in these individuals. Can I have the next slide, please? So we did we're doing some studies under Operation Warp Speed under a contract to Chad Roy here, and some of the program officers are at NIH are Gene Patterson, Clint Florence, Nancy Miller, COVTEN. This is a fairly large study. Also involves Tom Hope at Northwestern, and we're doing studies using monoclonal antibodies that were developed by Michel Nissenswag at Rockefeller University. The antibodies that he developed to SARS-CoV-2. So here you can see that this is animals were given infusions of 20 mg per kg or 6 mg per kg, 2 mg per kg, or control. And then you can see that the animals that got the highest doses are fully protected. At the lowest dose, you had a couple animals that showed a blip here, but for the most part, the kinetics show very nice control relative to animals that didn't get this treatment. I have the next slide. So very interesting is that we had also done a study to determine what would happen if we delayed the challenge. So this slide shows what happens in a few animals that were treated with these monoclonals and then were challenged 75 days later. And you can see that these animals here at the high dose, here and here, you can see that they're fully protected. There was one animal that showed a blip at day 2 post-challenge. And this is very different than the control animals, where you can see what's happening with the virus in these animals pre and versus post-challenge. Can I have the next slide? So, for conclusions, we've developed some animal models we think are going to be useful for vaccine, antiviral testing, and therapeutics, and we have many of these studies ongoing. Here, a subset of aged African green monkeys infected with SARS-CoV-2 recapitulated many of the disease manifestations, including ARDS and cytokine storm. The immunologic studies emphasize the importance of monocytes, macrophages, and cytokines produced by and then are affecting the migrations of these myeloid cells. SARS-CoV-2 studies in non-human primate brain suggest damage to blood vessels and the presence of microhemorrhage. And passive antibody studies suggest the potential for monoclonal antibodies as providing for significant SARS-CoV-2 prophylaxis. Next slide. So, in summary, Tulane, the National Primate Center here, we're providing platforms for the performance of vaccines, and we're doing, and therapeutics. We're doing these studies for Foundation Biotech and Pharma and NIH. We're doing studies in model development to look at the pathogenesis and or pathology of SARS-CoV-2. And we're doing some studies with passive antibody under Operation Warp Speed, and also multiple vaccine and therapeutic studies. We're also developing a national coordinating center for SARS-CoV-2 research for the NPRC system, and Nancy Haigwood will be describing more of this in her talk following, which will describe how this coordinating center and the network is working, so all our centers can work together. Next slide. As I mentioned, this really takes so many people that are involved in this work. I just want to point out Robert Blair, who was the lead on the pathology studies, Tracy Fisher on the brain pathology, Mana Fakari on the immune studies, Skip Bohm, our head veterinarian, also Laura Doyle is a veterinarian, Chad Roy on the Operation Warp Speed studies and his large NIAID contract. Also, we have a containment core, for the BSL-3 core staff, and these individuals here are really performing many of the studies in containment. And Angie Birnbaum directs biocontainment and operations and also quality control, and she leads also the containment and quality control efforts. Also, Chris Monjour, is additionally developed many of the studies for doing the viral loads that we're doing, and he's working in containment team also. Also, thanks to our NIH program officers, Jean Patterson, Clint Florence, Kuwait Dang, Nancy Miller, Sherry Hild, our program officer for the NPRCs, providing a lot of support for these studies. Michelle Nussenswag at Rockefeller University, Tom Hope at Northwestern, and some of the studies that we're doing on vaccines and others are really they're being initiated at New Iberia Research Center where Francois Villager is the director, and then these animals are shipped to Tulane for the challenge. Next slide. So our funding is from the base grant, P51 base grant, and our pilot study program. We got support from the Brown Foundation. That was our initial study with SARS-CoV-2 research. FAST grants to Monica Bakari and Tracy Fisher, NIAID and OWS funding to Chad Roy. And we have many additional studies in progress, which are supported by pharma foundations and academia. And we had significant investment very early from Tulane in performance of these studies. And next slide, please. So I'll stop here and entertain if there's any questions.
Perfect. I think we're gonna, just for the sake of time, we're gonna go through Nancy's first. And if you have questions for Dr. Rappaport, please be sure to click the Q&A box at the bottom of your screen and type in your questions, and we will receive it on our end. But to continue with this terrific lecture, thank you so much, Dr. Rappaport, for that terrific presentation and a lot of science, and that was fascinating. So Dr. Haigwood, I will turn it over to you.
Thank you so much, Naomi and Tinlu and Jay. Thank you for an outstanding presentation. I'm really happy to have a chance to talk to you for a few minutes about what the primate centers have been doing to coordinate COVID research. Next slide. And before I start, I wanted to remind the audience some of the reasons why non-human primate models are great adjuncts to other animal models. Next slide. Oops. Wrong way. Shows it's a bit of a busy slide, but basically the message here is that we've been around for more than 60 years working to model human diseases. And the rationale is that the very close genetic similarity is reflected also in all of the other body systems, reproduction, immune responses, physiology, as well as cognitive and metabolic responses, so that complex responses in response to pathogens that stimulate inflammation of the sort we've just seen can be modeled in primates. And because the animals are large enough and live long enough, we can do longitudinal studies that are typically not possible. And many times are not possible in clinical trials either, due to the need for very frequent sampling. And more recently, there's been an effort to develop non-invasive imaging at many centers, which allows us to do direct comparisons between human patients and animals, with animals surviving much longer and providing additional information. So it's incredibly exciting time to be doing primate research. And the most important aspect here is, of course, we can test potentially risky interventions, vaccines, and therapies prior to entering into the clinic. The primate centers, which I'll summarize in a moment, have primarily been breeding the Rhesus macaque. The Washington Center breeds the Nemostrina, or pigtail macaque, and then Southwest has for a number of years bred baboons. There are a lot of other species that are around, and obviously any animal model is tailored to the scientific question that is needed to address. Next slide. This is just a map of the US showing the location of the 7 national centers. And I'm from Oregon, of course, but I've worked closely with several different centers. And within the last 5 to 8 years, we've worked together as a consortium, a national network. We literally talk to each other every week. We have connections through about 20 different individual working groups covering everything from biosafety to you name it. And we have developed a couple of websites here in the middle. One is the nerdy website for those interested in doing research with us, where you find out what kind of facilities are at each center, what are all the publications, who's working there. And then the public-facing website is this one, the lower one, which is more oriented towards the lay individual. Our growth in the last, especially 30 years or so, has been stimulated by the discovery that primates were the only really good model for AIDS research. And we have been the beneficiaries of quite a bit of support from the National Institutes of Health, because those models recapitulate infectivity, immunity, and disease for AIDS. And so that investment is really what's paid off to where we are today. Next slide. And also shows an example of a few years ago when we were able to respond as a national consortium working together on Zika virus. This particular publication happens to come from Oregon, but I want to emphasize that this was the first time that the National Primate Research Centers had put together public website put together by David O'Connor at Wisconsin, where we shared real-time data. And because of this, and because of the existing expertise at these research centers in reproductive biology, in neuroscience, in imaging, in pathology, and infectious disease, it meant that we could quickly put together effective teams. And that's really the power of the centers and why I enjoy working at an NPRC so much. Next slide. So I want to turn now to what we can do to help solve some of the problems around the COVID-19 pandemic. Next slide. Back in the spring, Francis Collins set up ACTIV, the Accelerating COVID-19 Treatments, Interventions, and Vaccines, a set of groups at NIH. There were several different groups, including a preclinical group. And the goal of all of this effort was to bring together public-private partnerships to coordinate research strategies and really get to promising treatments and vaccines as soon as possible. And I think we're here today in large part or completely because of the tremendous expertise that was brought to bear on this from lots of different areas. I was invited to join this preclinical group along with Jay Rappaport, really to bring the knowledge of the primate centers, and then they asked us to work together to lead efforts at the National Primate Research Centers to coordinate our preparedness to contribute to this. I want to shout out to Joe Manetsky and John Young and Christine Colvis, who are the co-chairs of the preclinical group. They've been fantastic leaders, especially Joe, who's really driven us to thinking outside the box and moving quickly to solve problems and work together. Next slide. This is a diagram that is from a review article that Jay and I wrote with Judy Hewitt and other members of the preclinical working group this summer. And what it illustrates is the accelerated timeline that we have been attempting to develop. And this was our audacious goal at the start of the summer, was to do in 1 to 2 years what normally takes 10 to 15 years to do. And I think probably everyone on this webinar is very familiar with the careful phased versions of development of drugs and vaccines that typically take pauses between each step. And the goal here was to repurpose existing drugs and to use existing platforms and immediately insert SARS-CoV-2 genes so that we could test vaccines and therapies quickly in preclinical comparative animal models. And then as soon as there's a signal for positivity, start safety testing and then look for correlates of immunity and disease control. And as you know, large-scale manufacturing was done at risk. And as a result, we are on the brink of having vaccines here in the United States. We haven't been as fortunate with treatments, but I'm confident that the continuing work of ACTIV will yield that as well. Next slide, please. So this busy slide shows what we've been doing all summer, which is basically at the behest of the ACTIV leadership and in conjunction with an expert panel at NIH to prioritize research. We're working together, the primate centers, to provide some leadership, and that's Jay and I on behalf of the directors, and 3 of the operations leaders, Skip Boehm, Deb Fuller, and Koen van Rompuy, are all leading a group of 14 representatives from the National Primate Research Centers and a lot of other people who have basically put together what we call the COVTEN, or Therapeutic Vaccine and Therapeutic Evaluation Network. And this group then will work with the Tulane Center that Jay just mentioned to you. And all of the experiments that are done at the individual centers will then feed in samples and ideas. What the group has done this summer was a phenomenal effort, was to develop 42 separate SOPs, detailed experimental protocols that can be used for primate research. This is a feat, to me, was unimaginable at the start of the summer. It's involved more than 85 people. And we're incredibly pleased that we've been able to make progress on this. We also work together closely with people from other companies, NIH, other universities, experts like Ralph Barrick, And we have other working groups that are called Rigor and Reproducibility. And of course, we had a coronavirus working group. Members of those groups then all contributed, and we're now at the point where we can do this kind of coordinated research. Next slide. Just why would we do this? We believe we're uniquely qualified because of this deep immunology and virology expertise that really stemmed from the investments from HIV/AIDS, from the Office of AIDS Research and NIAID and ORIP over all the years. We have well-characterized primates and the facilities necessary to do this research at nearly every center. And what our goal really is to maximize expertise and resources, and therefore use fewer animals to achieve the goals by sharing controls, by sharing protocols, and be able to make comparative analyses across centers, as is done with human clinical trials. We're going to be using shared stocks. We're already using shared stocks, and you've heard about the samples already. You've heard about the protocols. Next two slides is a real quick look at what the field guide will look like. We've developed a field guide, which is the next slide, the beginner's introduction into if you want to do research. This is the cover, and the next slide shows the table of contents. So just to give you a sense of the soup to nuts, if you would like to do an experiment in non-human primates, what are the considerations? It's a lot to think about in terms of age, species, what kind of stock, how do you inoculate, what's the study design based on statistical analyses, what are the assays, what kinds of samples can you get, what's pathology going to bring you? And we're planning to share this with the public. It should be, I hope it will be posted before the end of the year. So next slide. What about resources? Well, I like to think of primates as being somewhat similar to the nursery business. You start out with a seedling and you don't really have a plant that is recognizable, usable for several years in many cases. And the same is true for primates. It takes 3 to 4 years for these animals to mature. We have a lot of complex algorithms in place to manage the species we have. For example, Oregon has over 5,500 animals or so. And we have about 2/3 of our animals are the breeding colony, and then we have animals for assignment. Because work needed to start right away, and we don't carry an excess of animals, any of the centers, because we have funded research and things are very tightly coordinated. We had to figure out how to fit this in. In a way, the COVID slowdown helped us because certain studies didn't start on time because people couldn't have access to their laboratories. And as Jay has mentioned, those centers that had pilot funding or seed money were able to go ahead with COVID studies right away. Oregon wasn't able to do that because we had some regulatory questions that needed to be addressed. And so our participation involved sending 100 monkeys to NIH for studies, some of which I will describe to you shortly. So it's really important that we do prioritize COVID research above everything else, but we don't want to stop our funded research. So it's a balance. That's why there are weekly meetings and lots of coordination in place. And we rely upon the NIH COVID-19 expert panel which approves all and gives a priority number to all NHP research. The goal here is to prevent someone coming in from the side with a bucket of money who would like to persuade you to do their study prior to one that might be a little more important on the national priority list. So next slide. So I'd like to turn to for just a few minutes to a little bit more data and talk about vaccines and antibodies. And this is a review that I highly recommend. Jay and Nathan et al. In Nature Reviews Immunology just recently published. It describes the vaccine landscape, which comprises 6 different kinds of types of vaccines that are currently under development, major platforms. And it's a compendium of the immune responses that are generated by each type of vaccine, and also a compendium of the trials underway. It's really a nice review. Next slide. I want to emphasize, because recently we heard an animal rights organization claim that primates were not necessary or even used to determine results for clinical trials, and nothing could be further from the truth. This is an example of the Moderna vaccine developed by Moderna and by the Vaccine Research Center. This is an article that was published in New England Journal. Barney Graham is the senior author, a beautiful study that shows in primates, in Rhesus macaques, how the humoral immune responses look in response to vaccines with 10 micrograms of the mRNA-based vaccine or 100 micrograms of the same vaccine. And I hope you can clearly see on the left, the binding antibodies are improved with the second immunization. But more importantly, perhaps, than the neutralizing antibody responses are greatly improved with the second immunization. And of course, the higher dose is giving a much higher titer, more than a log higher than the lower dose. Next slide. Similarly, when these animals were challenged with live virus, you could see both in the bronchoalveolar lavage fluid as well as in nasal swabs that while the controls had nice peaks at 2 days after infection, averaging over 10 to the 5th genome copies, that then declined in the bowel, but then were somewhat more persistent in the nasal swabs. Turning to the high dose vaccine, you can see 7 of the 8 animals had no detectable virus in the bowel, and I think 5 of them had no detectable virus. And even those with virus were significantly lower and cleared faster than the controls. The lower dose vaccine is not bad, but looks like the higher dose vaccine is better. Next slide. And then just last week, Dan Baruch and his colleagues published a pretty small study, but again, mirroring what Jay has just shown you with monoclonal antibodies. This was a study with passive immune globulin, IgG, purified from animals infected with COVID, and clearly shows that if you pre-load the animals with a high dose and then challenge them, there's no virus in the bowel or the nasal swab. A tenfold lower dose protects 2 out of 3, etc. Here are the controls. So it looks like a fairly high dose is going to be necessary of polyclonal antibodies. And similarly for monoclonals, by comparing the neutralization titers, you can get a better sense for how much exactly will be needed. Next slide then shows what happens in, again, a very small pilot with 2 animals per group, but very illuminating. I noticed that Jay, your study showed 3 days after. This is a 1 day after virus exposure, delivery of high dose, medium dose, low or no antibody IgG, and there's a diminution of the virus in the bowel and in the nasal swab that is clearly showing some activity, but only really in the high dose animals, and it's not very effective. So I think having powerful monoclonals on board would be much more effective, and of course better controlled since monoclonals can be reproduced in the lab. Next slide. So I think that's my last data slide, and almost this is my last slide. I just want to end with a few questions that we have been thinking about amongst the NPRCs as we all move forward with grants that we've applied for and collaborative research that we're working with NIH to do, and NIH and companies to do. And one of the questions, there's a hint for this first question in the data that I just showed you. One of the questions that's been concerning people significantly, certainly prior to the vaccine results in humans, was whether there was evidence for disease enhancement in the presence of pre-existing immunity as a significant concern for dengue virus. And I would venture to suggest, but the authors did not. So this is just my speculation that there was really no evidence of any enhancement of disease with these in this small study that I just showed you from Dan Baruch's group. Can more severe disease models be developed by perhaps ablating type 1 interferons. We have a very active discussion across the NPRCs to co-develop such models, and work is proceeding at Emory right now to work on this. And of course, we think we can contribute by modeling comorbidities. We have populations of animals that we have kept intentionally to do aging research. We have animals that have diet-induced obesity and are in pre-diabetic conditions. There are others that could be explored and are being explored to try to figure out why some people tend to have more severe disease, and can we model those in animals. Obviously, for the development of models that can help humans for therapeutic products, we'll need the detailed analyses that Jay just showed you of the cytokines and deep tissue analysis to really figure that out. But I think there's a lot that can be gained by doing additional primate work. Obviously, we can look at combinations of monoclonals, as he just showed you. And probably the most important question for me is, as we try to plan for future pandemics, is how can we do this strategic growth for the future to be ready for the next outbreak, so that we're not caught off guard as much as we were. We did better than we did with Zika. On the other hand, this is a terrible pandemic that we have not controlled yet, and we still have quite a lot more work to do. And we are in deep discussion as to how we could best work with partners to expand our research, because we think we have a lot to offer. So I'll end there and thank my colleagues here at Oregon and all of my colleagues at all the NPRCs for their suggestions.
Perfect. Thank you so much, Nancy. No, that was a great presentation. I really liked the analogy with the nursery business and just your emphasis on planning and how important that is to make sure that everything is working together. Both of you, thank you so much, and we really appreciate your expertise and your time to speak with us that are living day by day, not really sure what the status is out there. So again, we're going to move now into a Q&A session briefly. Just as a reminder, if you want to ask a question, just click the Q&A button at the bottom and type your question, and we will see it on our end. For those that may have a question for me or anyone here at FASB, this is my name and my information. Again, these slides will be available, so don't worry about that. The recording will also be available, but please feel free to email me questions that I can relay back to the speakers. I'm happy to do that. So I will stop the share now, and let's see what questions we have. Okay. Question for Dr. Rappaport. This is quite a bit of a long one, so bear with me and I will try to shorten. As you know, diabetes and dysglycemic states seem to be critical for the aggressiveness and risk of death in COVID-19 humans. How did the African green monkey that became with ARDS develop the cytokine storm, have their metabolic phenotypes such as glucose levels? Was this an influential factor to have developed ARDS and COVID-19 severity?
Well, it's a difficult question. I don't believe that any of the animals that did have ARDS had evidence of diabetes. I think that there may be some increase in glucose in the animals that did get ARDS, so I have to go back and look at that, but that's something that I recall. But this is a very interesting question, and something that is being looked at the California Primate Research Center now, with using animals rhesus, which normally don't get a severe infection like this, and infecting diabetic animals with SARS-CoV-2, and to see how that would affect. But I think that's going to be very important. It's unclear how diabetes predisposes to SARS-CoV-2, but SARS-CoV-2 seems to make diabetes worse. At the same time, the non-classical monocytes are known to be increased in diabetics, and I showed you that there's some, at least from our studies, there's some trafficking of those cells into the lungs. they may be providing, some mechanism of pathogenesis. The further studies needs to look at that to fully confirm that. But it may be that what's going on in diabetes and expanding non-classical monocytes may have an effect in predisposing for SARS-CoV-2 infection.
Wonderful. Okay. Another question we had is there's just been some news circulating about the demand and access to non-human primates at primate centers. Of course, we think will eventually exceed the number of available animals. And Nancy, you mentioned the NIH expert panel kind of prioritizes which studies need to happen first. But what else are the centers talking about that you can shed some light here? And how can we, as an advocacy organization, as scientists, help to spread the word of the importance of access to these animals?
Great. Well, I think that I appreciate your offer to continue your important advocacy. We very much need that. I would say everyone who's interested in the development of, and use of animal models for human diseases, especially this one, can help us by educating other people, including your colleagues who may have misinformation about the role of primates in development. Obviously, they complement other animal studies. As far as the difficult problem of running out of resources, I would say we are at our nearly at our limit, if not at our limit, certainly at some centers. We do share resources across the centers very actively all the time, but in order to really overcome this bottleneck, it's going to be important to do long-term planning. And I want to call out the Office of Research Infrastructure Programs, ORIP, which had commissioned a couple of years ago a study on needs assessment. And Sherry Hild, the program officer for our P51 grants, led that effort. It was an external effort, but she spearheaded the activity. And I think it was very timely at the time. And the messages that were contained within that NIH report are certainly the ones that we need to pay attention to right now. Right. We need to enhance and increase breeding and find ways to support the animal care staff to support all those additional animals.
Right.
If I could add, there is quite an effort beginning now with ORIP and providing funding to expand the breeding colonies. And it's just not more animals, but it's also the infrastructure, the field cages, and other support, the staff. So there is an effort now to expand the size of the colonies and make more animals available specifically for something like this pandemic. And also there is another effort that we're doing. One of the reasons we're coordinating so tightly across the different NPRCs is so that we can perform these studies in the same way, so that we can compare data from one study to another, regardless of where they were performed. And we're actually going to be sharing control groups so that each experimental group doesn't have its own control group, but we can share controls across the center. And that will also limit the number of animals that we'll need to use in these studies.
Makes sense. So on the topic of advocacy, another question we had was, how do you explain the importance of animal research, and particularly non-human primates, to someone who may not have a scientific background, and is not too familiar with some of the jargon and know what that is? What's the best way to approach that?
I'd recommend going to the public-facing website that we developed as one first approach. There are other groups that have developed talking points that are really excellent. Both Americans for Medical Progress and NEJM are really outstanding groups, and each of the primate centers has an outreach program for the public. The real bottom line is that we accelerate the development of products for humans to help improve human health. And we have so many good examples. I think when you're talking to a scientist, you would choose a different perhaps different kind of example, but when you're talking to, for example, an older individual, we can talk about macular degeneration and how we've developed AAV vectors to treat the progressive blindness in the eye. That there are so many important efforts ongoing right now and so many good examples. So it's going to take, like science discussions in the United States in general, a lot of us a lot more time. We need to get out there and explain what we do and why we think it's important.
Right.
Yeah, that's a good example you gave about macular degeneration, and mice actually don't have macula. So this is one of the reasons, actually a really good example in a specific disease where non-human primates are the model.
Right, right. Yeah, that's a good point. I think size matters, in relation to the disease that you're studying. And then,
Yeah, I'm seeing a
humans, similarity to the human immune system is very important.
Mm-hmm.
Wow.
Sorry, Naomi, I was seeing a question from the chat, or from the Q&A.
Yes, did you want to take that one about studying a novel pathway that could be important? How do we access samples to see expression of these molecules and discuss experiments to assess these pathways in the non-human primate models, both in vitro and in vivo?
I was going to send that to Jay, since he's really trying very hard to set up this coordinating center, which has the exact plan to do that.
Right.
Yeah, I mean, if you have a novel pathway, and you think it's really important for COVID, it'd be important to generate in vitro data, and let's say if it's replication, if it blocks the virus life cycle, maybe that's something you could do in vitro. If it's something that where you're looking at the immune system and you want to modulate the immune system, well, maybe there's something that you need to test in an animal model. Right now, there's an expert panel at NIH that is really determining the prioritization of animals and for all the SARS-CoV-2 studies. So there is a website that you can submit your idea, and then you get feedback also based on a priority rating. Now, if it's something that there isn't already, they're prioritizing the compounds that already are approved for human use or have some novel pathway, and maybe there's some existing data in small animals. If you don't have small animal data, then it would be important to try to get some, maybe in a mouse transgenic model like the COVTEN or the hamster model. And then that could help get prioritization to get access to non-human primates. Unfortunately, there's just not enough animals available to test everything right away in non-human primates. And I think, how important that resource is. I think it's also important to test the strategies that have the most promise and are the most advanced, in the species as well. So I would encourage you to sort of look at those steps in vitro, in vivo, small animals, and to look at the submitting your compound, your pathway to the expert panel.
That's a great idea. And you said there was a website where you can include that.
Yes.
Okay. Okay. So go ahead.
We're out of time, but I think the question was also specific access to tissues. And
tissues. Yeah.
I might want to just contact some of the authors that are part of the studies that have been ongoing so far to see if they've already looked for these markers, or if they might be able to provide tissues if you have the right containment facilities.
Yeah, we have, I mean, if you want fixed tissues, that's no problem in getting them out of containment. And we have a program where we provide tissues to people all over the country and all over the world, for example. So, we've been entertaining sample requests. If you would like to look at specific tissues, if you want to look at lung or wherever you'd like to look, we can make specimens available. And all the NPRCs have similar programs in making specimens available.
Great. And the last question, I know I want to be respectful of everyone's time, is the website that Jay, you mentioned. Is that something I can quickly go to and insert here? Or how about I do this? Along with the slides, I will include that website and we will have that as a resource for folks that are interested.
Okay, great.
Wonderful. Well, thank you both so much. I know we're a couple minutes over, but you guys have been terrific. We really appreciate all your working around the clock, but taking some time to speak to us about all your incredible work. We appreciate it. And I hope everyone enjoyed this webinar, and I have a wonderful and safe holiday season.
Thanks so much. And you too, Naomi. And I hope everyone has a great holiday and New Year.
So much.
Thank you.
Bye.
All right.